Hello everyone, welcome to sqadia.com. Today we're discussing cellular energetics, and after we have discussed how the proton motive force is being produced, now it's time to discuss how the proton motive force is being harnessed and how it is being utilized in several different processes. So we are going to start off with the discussion for the F1 need for the F1 subunit for the ATP synthesis, because apparently it does not have the channels for the proton half channels to move the proton from outside to the inside. So the question arises, what is actually the need for the F1 subunit of the ATP synthase for the synthesis of the ATP? So we are going to tackle this first question in when we're going to start off with this section. Then we are going to start off with the discussion of binding change mechanism for the ATP synthesis, moving towards the phosphate, ATP and ADP transport system. In this system, we are going to study the antiporters mechanisms for the transport of ATP and the phosphate groups as well that are required for the production of ATP. So starting off with the discussion of need for the F1 for the ATP synthesis. So initially what we have, the inner mitochondrial membrane, it has the F0 unit and it has the F1 subunit, which is represented as a head, and F0 as a tail. So what happens when the inner mitochondrial membrane is being treated within ultrasonic vibrations, they are going to be converted into the small submitochondrial vesicles. Now these small submitochondrial vesicles, they are capable of two things, both electron transport chain and the ATP synthesis. Now, what we do is to understand why, if there is a need for the F1 for the ATP synthesis. So what we are going to do, we are going to mechanically agitate. It means that we are going to use force to separate the F1 subunit from F0 subunit, as shown in this particular portion. As a result of mechanical agitation, the F1 subunit is being detached with the F0 subunit. So the membrane vesicles at this stage are only capable of electron transport chain, but the experiments have shown that they are not capable for the ATP synthesis, but only for the electron transport chain, because surprisingly or supposedly only the F1 particle has been removed. So it means that F1 plays an important role for the ATP synthesis and not for the electron transport chain. So what happens in the very next step, because they have been mechanically removed, they can be mechanically combined as well, and we are going to reconstitute these vesicles by the reversible step. What we'll do over here, we are going to combine again the heads with the F1 subunit with F0, and then we are going to see if the reconstituted vesicles are capable of both electron transport chain, electron transport, and ATP synthesis. So it is seen that the reconstituted vesicles are in fact capable of both the running down the electron transport chain and the ATP synthesis as well. So it means that the F1 is primarily specific for the ATP synthesis, but is not specific for the electron transport chain reaction for the transport of the electrons. So talking about the binding change mechanism for the ATP synthesis, we have this diagram is basically showing the upper portion of the ATP synthase. If we are going to look at the upper portion in this particular manner, so we are going to see ATP synthase looks like somewhat like this. So it has three phases. The phase O, which is basically in the nucleotide binding domain. We have the L phase and the T phase. So initially, the O is being free. Of course, it is going to cater the binding of the ADP and the inorganic phosphate. This binding is going to cause the morphological changes, energy that is going to cause a production of the rotation of around 120 degree. So as a result of rotation of 120 degree, of course, it is going to be moved. The L position, of course, which was initially here, is going to be taken by the O position. O position is being taken by T, and T position is going to be taken by L. So what happens the as a result of energy, where ADP plus inorganic phosphate is present, we have at O site the ATP is present because it has shifted its site. Initially the ATP was being catered by T. Now ATP is being catered by O because there was a shift rotation of 120 degree of the gamma subunit, of course. So the L unit is now catering the ADP plus inorganic phosphate. This plays a very important step because over here you can see that there is the formation of ATP in T site. It's important to consider that the ATP is going to be formed in the T site because the ADP and the inorganic phosphate were entered. So there is no need for the energy in this particular case. Just the inorganic phosphate is going to be added as a result of energy movement, as a result of a 120 twist in the gamma subunit. So what happens in this case, the ADP is being produced with the yield of water as well. So when this is being produced, the O site would get empty. Now it means because the ATP was produced in this step as well, so the ATP would be removed and the O site would be ready for the next binding of the ATP. So this particular figure is the exact same of this initial figure, except for the case that it is being rotated 120 degree. So what happens in the very next step, the energy will further cause the rotation of the ATP molecules, where the ATP is being released. ATP is going to then cater into the ADP, the L side and the T side as well. So this process is going to continue, resulting in the production of ATP yielding the water molecule as well. So the phosphate and the ATP or ADP dependent transport system, we have the inner mitochondrial membrane as shown. We have a concentration gradient down the matrix. So, and we have of course the two-thing membrane electric potential as well. Both of these combined would serve as a proton motive force. So there is a translocation of a proton against their concentration gradient during the electron transport, when the energy from the two electrons are being utilized for the movement of protons outside. So we have a phosphate transporter, we have an ATP antiporter molecule. All of these combined is going to help in the production of the ATP. So we are going to see that the OS that has been removed from inside to the outside binds with the some of the protons that has been translocated out against the concentration gradient would combine to form water. And similarly, this antiporter, the phosphate transporter, which is actually the antiporter, would simultaneously move the phosphate ions from outside to the inside. Now it's important to now let's pause for a moment because we have seen that the phosphate ions have moved inside. Now we are going to see in the case of the ATP and ADP antiporter. So what happens, they are going to cause the movement of the ADP from inside to outside and ATP4 minus from inside to the outside. So we have two things, ADP3 negative and we have for HPO4 minus 2. Both of these are going to combine, resulting in the formation of ATP4 negative plus OH negative, which is then going to cause the release of of course the electron, resulting in the yielding ultimately of H2O and ATP molecule as well. So in this section, we started off with the discussion of the need for the F1 for the ATP synthesis, and then we are going to see the binding change mechanisms of the ATP synthesis, and then we are going to see the phosphate and the ATP or ADP transport system. So that was all for this section. Thank you for watching sqadia.com.